Dear students, let us learn about oncogenes and tumor suppressor genes in this lecture. Let us start with the introduction. Cancers originate in cells mostly from inherited and somatic mutations of genes that control cell proliferation, differentiation, and survival. Such types of genes include two main classes of genes, that is, proto-oncogenes and tumor suppressor genes. An oncogene is the gene which can induce cellular transformations causing cancer, while proto-oncogenes are normal genes that can become an oncogene due to mutations or increased expression. In tumors, the increased expression of the oncogenes leads to uncontrolled activity of the oncogene-encoded proteins. On the other hand, the tumor suppressor genes aid to inhibit cell proliferation and tumor development. In tumors, therefore, the tumor suppressor genes are usually lost or inactivated, resulting in uncontrolled cell proliferation. Let us now take up oncogenes. Although the role of oncogenes in cancer were first studied in tumor viruses, further studies investigating the molecular mechanisms of cancer led to the identification of several other cellular oncogenes, which played a role in the development of non-virus induced cancer. The important aspects of oncogenes can be discussed under the following sections. The first are the retroviral oncogenes. Several oncogenic retroviruses have been reported to cause cancer in many animals such as chickens, turkeys, mice, rats, cats, and monkeys. These retroviruses usually contain one oncogene, in some cases two oncogenes. These viral oncogenes are not required for normal virus replication, but they induce cell transformation. Some retroviruses may contain the same oncogene, but many types of oncogenes have been identified from the retroviruses, and the proteins encoded by some of these oncogenes are components of cell signaling pathways that stimulate cell proliferation. One example of a retroviral oncogene is the SRC oncogene of Rous sarcoma virus or RSV, capable of inducing tumors in birds and also transforms fibroblasts in culture. Next are the human oncogenes. When the origin of the retroviral oncogenes were studied in humans, it was discovered that they arose from closely related genes of normal cells called proto-oncogenes. The retroviral oncogenes, for example, SRC, RAS, and REF, encode proteins that play a role in the signal transduction pathways controlling normal cell proliferation. When a proto-oncogene is incorporated as an oncogene in a retrovirus, its expression is increased as it is transcribed under the control of viral promoter and enhancer sequences. Such increased expression may lead to the conversion of the proto-oncogene into an oncogene. In addition, the proteins encoded by the oncogenes may differ in structure and function from the normal homologs, which may result in unregulated activity of the protein, resulting in transformation and cell proliferation. In addition to the retroviral oncogenes, many other cellular oncogenes have also been identified that differs from the corresponding proto-oncogenes by point mutations. In human tumors, the proto-oncogenes can be converted to oncogenes mostly by point mutations, but several other mechanisms such as chromosome translocations and gene amplifications may also frequently lead to the generation of oncogenes. First, let us see the oncogene activated by point mutation. Some of these human oncogenes are homologs of retroviral oncogenes, while many other new oncogenes were discovered in human cancers. For example, the most common oncogene in human tumors includes the human homolog of the RAS-H oncogene of Harvey sarcoma virus and are represented by three closely related members of the RAS gene family, that is RAS-H, RAS-K and RAS-N. The RAS oncogenes result from point mutations in their proto-oncogenes, resulting in single amino acid substitutions in the protein encoded by the oncogenes, and such amino acid substitutions lead to unregulated activity of the oncogene proteins. The RAS genes codes for guanine nucleotide binding proteins that play a role in mitogenic signal transduction from a variety of growth factor receptors. The activity of the RAS proteins involves the alternation between active or GTP bound and inactive or GDP bound states and point mutations leading to substitutions of a single amino acid, usually at position 12, 13 or 61, maintains the RAS proteins constitutively in the active GTP bound conformation. These oncogenic RAS proteins nullify GTPase activating protein or GAP, which stimulates hydrolysis of bound GTP by normal RAS. As a result, the intracellular activity of GTPase decreases and the oncogenic RAS protein remains in the active GTP bound state, causing unregulated cell proliferation. Next are the oncogenes activated by translocation. An example of a human oncogene activated by chromosome translocation is the CMYC oncogene, causing malignancies of antibody-producing B lymphocytes such as human Burkitt's lymphoma and mouse plasmacytoma. In Burkitt's lymphoma, a fragment of chromosome 8 is translocated to one of the immunoglobulin gene loci on chromosome 2. The CMYC gene codes for a transcription factor that is normally induced in response to growth factor stimulation. However, when the translocations cause the insertion of the CMYC proto-oncogene into an immunoglobulin locus, it results in uncontrolled expression of CMYC gene, which drives cell proliferation and contributes to tumor development. Another example of an oncogene formed by translocation is the translocation of ABL proto-oncogene from chromosome 9 to chromosome 22 in chronic myelogenous leukemia. Next are the oncogenes activated by gene amplification. Gene amplification is another distinct mechanism by which oncogenes are activated in human tumors, and amplification of the oncogenes may lead to rapid growth of tumors and increase in malignancy. For example, amplified copies of the NMYC gene are present in rapidly growing aggressive tumors of neuroblastoma, which is a childhood tumor of embryonal neuronal cells. Gene amplification has also been reported in the oncogene ERBB2, which encodes a receptor protein tyrosine kinase that is related to progression of breast and ovarian carcinomas. Next, let us take up the tumor suppressor genes. Tumor suppressor genes repress or inhibit the cell cycle or promote apoptosis. The proteins encoded by the tumor suppressor genes act by stabilizing the genome, providing DNA repair factors, and by inhibiting other activities such as cell cycle progression, mitogenic signaling pathways, pro-growth programs of metabolism, and angiogenesis and invasion and metastasis. The classic tumor suppressor genes are generally recessive at the cellular level, and both alleles are typically inactivated in tumors, while inactivation of a single allele generally increases tumor susceptibility. There are many tumor suppressor genes, and each tumor suppressor gene have a complex mechanism of action. Some common tumor suppressor genes are described below. The first one is retinoblastoma or RB. The retinoblastoma gene is a tumor suppressor gene that encodes the RB protein. The retinoblastoma protein binds to a transcription factor E2F, which results in blocking the transcription of genes necessary for DNA replication. It arrests the cells in the G1 phase of the cell cycle and prevents the cells from entering S phase of the cell cycle. Next is the TP53 tumor suppressor gene. The TP53 is another tumor suppressor gene, and it is also known as the guardian of the genome, as it plays major role in inducing apoptosis when DNA repair is not possible after identifying DNA damage and pausing the cell cycle progression to give time for repair. The TP53 gene codes for a protein called p53, which also prevents the cell from entering S phase by inhibiting cyclin-dependent kinase 4 or CDK4. When DNA damage is severe, the p53 protein induces apoptosis by activating the BAX gene, which codes for a pro-apoptotic protein. p53 protein also inhibits the gene BCL2, which is an anti-apoptotic gene, and it stimulates the release of cytochrome C from the mitochondria. Cytochrome C further activates caspases within the cell, leading to the eventual degradation of the cell. Loss of p53 may lead to continued cell replication despite DNA damage and failure to activate programmed cell death. The next is the phosphatase and tensin homolog or PTEN gene. The phosphatase and tensin homolog or PTEN gene codes for a PTEN protein that negatively regulates the phosphorinositide 3 kinase or P13K AKT and the target of mTOR signaling pathways, which play important role in cell proliferation, cell cycle progression, and apoptosis. The PTN protein also plays a role in stabilization of the genome and to control migration, adhesion, and angiogenesis. The next is the neurofibromin 2 or NF2. Neurofibromin 2 or NF2 is another tumor suppressor gene that encodes for neurofibromin 2, also known as Merlin, which acts downstream of E-cadherin or epithelial cadherin that mediate cell-to-cell contact and regulate contact inhibition or the inhibition of proliferation of normal cells when they come into contact with neighboring cells. The epithelial cadherin binds to beta-catenin, which is a key component of the WNT signaling pathway, and the binding prevents translocation of E-catenin to the nucleus, preventing it from activating transcription of pro-growth target genes. Next are the BRCA1, BRCA2 and PARP1 genes. BRCA1, BRCA2 and PARP1 are tumor suppressor genes that encode proteins involved in the repair of DNA damage, and they act to prevent the cell from continuing the cell cycle when there is DNA damage in the cell. The proteins encoded by BRCA1 and BRCA2 play a role in the homologous recombination pathway for repair of double-strand breaks. On the other hand, the protein product of PARP1 play a role in the repair of single-stranded breaks in the DNA. Next is the APC gene. The APC gene is a tumor suppressor gene that codes for adenomatous polyposis coli, which blocks the WNT signaling pathway affecting cell migration and cell proliferation. The APC protein also plays a role in apoptosis. Now coming to the conclusion. Although the development of cancer is a multi-step process controlled by accumulation of damage to multiple genes that result in increased proliferation, invasiveness, and metastatic potential of cancer cells, the activation of oncogenes and the inactivation of tumor suppressor genes play important role in tumor initiation and progression. While the oncogenes drive abnormal cell proliferation by harboring tumor-activating mutations, the tumor suppressor genes become inactivated in cancer. Thus, the uncontrolled proliferation of cancer cells is the result of mutation in several genes that normally regulate cell proliferation, differentiation, and survival. Thank you.